Howdy. I think I might just go straight to the point while I'm here. All right. Today I'm going to be talking to you all about some things that we have done a few years back, 2015. We did a study with methionine supplementation sponsored by one of our companies in Belgium. So I'm going to be talking about that study, how we did that study, and a few things in that study and some of the data that we collected and how we collected that data. So the topic of today is going to be on the tolerance to increased supplemented levels of methionine in older adults. And these were all healthy men that we recruited to come participate in this study, and we needed just 12 people to complete the study, and there were 6 men and 6 women, but we went a little bit higher in case there were some dropouts. All right, this is going to be the outline of the discussion. So I'll have some introduction about what we are talking about, the objective of the study, and then the methodologies, how we collected data and how we performed that study. Some of the results, I'm not going to talk about it all today. I have a colleague that will be presenting, which she is in the same group in which I am too, so she's going to be talking about that section of the study. She was also involved in collecting some of these data. Then some discussions, and then conclusion, and then what we thought of doing after we had conducted this study in healthy adults. So we started thinking about doing the same thing with the sick people, especially those with liver disease. So today we are going to focus more on the healthy older adults, and then some questions. All right, a little thing about methionine. Maybe some of you know or you don't know, methionine is an amino acid that is very essential or very important for every human being, because it is one of those amino acids that is really necessary for protein synthesis, and it's also the start codon to make proteins. So with that being, it's also a precursor for many other amino acids. We'll get to the metabolic pathway of methionine and you'll see what I mean. So there are some amino acids that can be derived from the production of methionine. When you supplement methionine, it leads to the production of so many other different amino acids, and that's because it's one of those only amino acid that has a sulfur group. And you have amino acids like cysteine and the tripeptide like glutathione that also has sulfur groups in there. And we all know the importance of that S group that is in the methionine acid. All right, so you know, as we old or as we age, there's some changes that occur in the body, and these changes are usually affected by age, especially our muscle mass, especially our health. Everything starts to change or goes down depending on your lifestyle. If you were very active growing up, or if you stay active, things will still change, but in a very minimal level. But if you are very sedentary, then things will change very drastically. And that's why in our lab we focused more on comparing COPD people or sick people with healthy people, so we can actually try to make differences and see how all this metabolic pathway they function. And one of the primary precursors of methionine is homocysteine. So we are going to talk a little bit about homocysteine, because you can only get homocysteine through methionine in that pathway. When we get there, we'll see how it's derived and some of its beneficial effects and those things that happen along. All right, what the objective of this study, why we did this study, we wanted to look at the no observed adverse effect level of methionine supplementation, and we also wanted to look at the low observed adverse effect level of this same amino acid. So we wanted to look at, if you put it in one word, it's toxicity level. If how much of methionine would you supplement for it to become toxic for human consumption? And also, how much will you have to supplement for the methionine to be able to do its job, to take care of all the other things that it does in the body? All right, so going to methodologies. All right, when we came out with the protocol of this study, we also decided to come out with some reasons how we have to recruit people to participate. So we had some inclusion-exclusion criteria, and some of them are listed above. So we wanted healthy male and females to participate in the study between the ages of 40 and 79. So we believe that those are the people that we wanted to see. And then this study was going to be lasted for 4 hours every time. So they had to come in at least 13 visits. And then it was going to be 8 long visits and 7 short visits. So I'm going to show you this on a little diagram that I made, or a time clock for the study that I made, so you can see. And there were also some exclusion criteria. You know, when you design a study, you want to make sure that people are eligible to participate. And so you go by selection, and those selection will be based on your inclusion-exclusion criteria. What are those things that will make a subject not eligible for participation? So I have a list of all this, and maybe I'll leave the slide so you can forward to the students later.
Do you mind enlarging the screen?
Oh, I was over here. I think I tried that it didn't.
The icon on the bottom right there.
That one or this one? Okay.
Get close.
Yeah. Which one?
This
This
guy
one?
right here.
Oh,
Got it. There you go.
yeah, that's better.
A little bit.
All right, so as you can see, some of those criteria, and that goes as far as seeing if the subject is unwilling to participate by not signing the consent form. All right, so we had 3 types of visit that the people that we recruited in the subject, in the study, when they came in, we would accept them in the study. Number 1, we had a screening visit not lasting more than an hour, so we could just use that time to find out if these people were eligible or not eligible. And then when we found out that they were eligible, then we then scheduled them for their study days. As you can see, 13 study visits, 8 of them were long visits, and the other ones were short visits. And I will explain to you what a long visit, according to our research, means, what we do during those 4 hours when they came in. All right. During our study days, we did do some measurements. And some of those measurements, some of us in here already know. We did body weight measurements, we did BMI measurements, and then we also did body composition. And I'm going to break this down in the results so you can see the type of data that we collected during the body composition measurements. All right, this is a timetable for the different visits that we did. Do you know if we have a pointer? I could show some few things. All right, so these are the different doses that we actually supplemented, and each dose lasted for about 4 weeks. So as you could see, we started with an intervention of 10 milligrams. We did that for 28 days, and then we gave them a 2 weeks time to flush out, and then we increased the dose to 25 milligrams. That was based on body weight. And then we did the same thing. We increased to 50 milligrams to 100 milligrams. We never went up to 150, because once we got to 50, we started seeing some things that made us to go to 100 and see if we'll see the same effect or an increased effect or an increased toxicity effect. So we decided to stop at 100. So we did not exactly give 10, 25, or 50 milligrams. We gave something approximately, because we calculated the dose based on body weight. So we had a little Excel sheet. When you come, we take your weight, we plug it into the Excel sheet. It gives us an automatic dose based on your weight, because we believe that a person that's 70 kilograms and a person that's 50 kilograms will not take exactly the same dose, but the dose effect based on calculation will be equal. Okay. So, as you can see on this slide, we did offer them free methionine supplementation for 4 weeks, where they started and ended with a long visit during each time when they came in. When we did the measurements, we started with a screening visit as usual, And then if you see here, you see 9.2, 22.5, 46.3, and 91. Those are the appropriate or approximate doses that we gave based on calculations depending on body weight. And all these capsules, we ordered the powders, and then we made the capsules ourselves based on some other protocols and other papers that we read, how to supplement and how to do an intervention study. All right, this is the timeline of data collection or sample collection for 4 hours. So when that subject came in, we have amino acid compounded that we infused or injected about 8 mils of a combination of different amino acids, including methionine and homocysteine. And then we collected blood samples for a period of 4 hours. So what that means is when they came in, we normally check if they are fasted prior to the night before. And then we insert an IV with a little catheter in there and leave it in there for the period of 4 hours. So we started collecting blood. As you can see, the first hour, it's a little bit continuous. We collected blood at there was a baseline, there's a 2 minute, there's a 5, 10, 15, 30, 40, until the 60th minute. And then after the 60 minutes, from the 1 hour period to the 4th hour period, it was every 30 minutes. The reason for doing so is because we also wanted to compare blood sample time points with the background. So we take each time point and compare with the background and look at the effect and look at the metabolic pool of these amino acids during that period of time. Because things could be happening, changes in the conversion of methionine to homocysteine, to all the other things that we were trying to look at. All right, this is just the metabolic pathway of homocysteine, how it can be converted in the body. If you take methionine and then it's going to be converted with an ATP molecule to S-adenosyl methionine, and then it's going to go to homocysteine until we get to glutathione, because like I said earlier, it's one of those amino acids that has sulfur, and it's going to help in the production of amino acids that carry sulfur in that peptide. Thank you, sir.
Here's that's a portable mouse if you want to travel. So.
Okay.
All right.
Let's see. Okay. All right, it's good. All right, we did some biochemical analysis with all the samples that we collected during that study period. So first of all, the way we collected the blood, you guys might want to ask the question, how did we keep blood flow for 4 hours? So we have a little component, a little machine that we use. We call it a hot box. We place the arm in there with the IV, so it keeps the hand warm, and it also helps us to get good blood flow and help us to get arterialized blood through the vein. So it's easy to get a good sample that we could analyze. And those samples were immediately placed on ice and taken into a BL2 lab where it was centrifuged at 5,000 for 8 minutes, so we can collect the plasma and transfer in different Eppendorf cups or matrices, and then put in -80 for later analysis. And then we added some different types of solutions in there for deproteinization, and also some of the samples were collected in SST tubes, the little lavender tubs and the tiger tubs, so we can ship some of those for immediate analysis on those things that we wanted to look at based on time. All right, as you can see, this are some of the parameters that we measured, those things that we sent to the lab. So the top part are some questionnaires that we asked the study subjects during our study period. Some of these is related to mood, some of these is related to toxicity, some of these are just related to the way depression, cognition. We also did that so we can see the effect of mood and memory and behavior during that time when they were consuming this methionine supplements. And the rest of these down here are just some of the tests that we measured through different labs, because we had to ship on a daily basis when we did that study, like homocysteine in plasma. we send it to Quest with Quest Diagnostics, if some of you know that. And some we send to LabCorp. It's also another lab. It's based in Houston, so they can analyze those samples. And what I did here was just based on cost effective. So some of these samples all of them could be run in one lab, but some cost more in other labs. So we try to manage based on the budget that we had, so we can effectively do everything timely and on our budget. All right. We also did some statistical analysis with all of these stuff, and we mostly did the two-way repeated measure analysis, or ANOVA. And our P level was less than 0.05 as everybody else. All right, some results. All right, changes in body weight. So technically, we did not see any changes in body weight. There was no significant changes, and that is something that we knew it was going to happen, because we did not supplement or do any other thing that will affect body mass or body weight changes. So you can see here between the 10 milligrams, the 25 and the 50 and the 100, it's approximately the same. So very little changes, but not significant changes where we could report a P level. And then based on body composition DEXA measurement, we did the fat-free mass measurement, and we also did the fat mass measurement. And between the 2 weeks to the recovery time, we did not see any changes. So here we have the stat initially, and then after 4 weeks, then after 4, before they came to the next level of intervention, we gave them a 2 weeks recovery. So when they come in every time, we always did body composition or DEXA measurement. So we want to make sure and check to see if there were other changes. And one of the other things that we did was we did follow-up calls on a daily basis with these subjects, so that we could ask them some questions, how they were feeling, how they were doing, and if they had anything like nausea or they were vomiting, because as the level of the intervention increased, there were certain things that would happen. So we did those follow-up calls to make sure we monitor these subjects in case of serious adverse effects. All right. So this is the supplementation that we supplemented with. We initially thought we were going to give 10 milligrams, 25, 50, and 100, as I said, but we did calculations based on body weight. So an approximate, we're using standard error measurement. So we had 9.3 for the stat, and then 9.2 after 4 weeks, and then at the recovery, they didn't consume anything to go to the next level. So when they came to 25, the approximate was 22.5. For 50, it was about 43.3, and for 100, it was about 91 milligrams per body weight. All right. So we did know that increasing would increase homocysteine concentration. That's one of the things that we were focused on, to look at homocysteine concentration, because it's related to toxicity, and it's also related to the methionine that we supplemented at baseline. And then at 10 milligrams, there was really no change in concentration, but we saw increasing concentration when we went from 10 through 25 to 50 and to 100. So the homocysteine concentration increased significantly, especially between the 50 milligram supplementation and the 100 milligram supplementation. All right, this graph here, it's also kind of showing the same thing, but at this time we wanted to measure each individually to see how the changes affected the subjects. So if you look at here, we have 4 different levels: the 9.2, the 22, the 46, and then the 91, or say 10, 25, 50, and 100. And we technically did not see any change with the 10 milligrams for a whole 4 weeks. And when we supplemented with the 25, there was a little change, so a little increase, and then came back to normal during recovery. Then when they came back for the next 4 weeks after their 2 weeks of flush with the 50 milligrams, there was also some increase, but not really significant. And then during recovery, it went back to normal. And then when we supplemented with the 100 milligrams, we saw a drastic increase in concentration and also increase in the level of the methionine or homocysteine in the system. And then it stayed very high until when they got to the recovery, everything went back to normal. And that's where we actually observed our significant level of supplementation. All right. Some of the panels that we actually did measure, homocysteine was one of them. And after doing our statistics, we found that the time was also significant because they had to do that within a period of 4 weeks. We also found that the dosage was significant because it was very high. We actually doubled from 50 to 100, so that's actually multiplying it by 2. And then there was also an interaction effect with significant level of 0.001. And then we measured the glucose level to see if it also affected. So we really did not see anything except at dosage, which was close to significance. And I think the highest we had was about 139 for some of the subjects. All right, carbon dioxide. Time and dosage and interaction. And then the liver panel with the AST, we also saw a time effect and we also saw a dosage effect. And then with here with the monocyte on the complete blood count, we did see a little bit of change, but it wasn't close to significance. And then the lymphocyte, the white blood cells that are supposed to help with so we saw a dosage effect. And after 4 weeks, there was really nothing. It was just during the baseline. Then with the monocytes, the same thing. So mostly all the things that were related to the complete blood count, we saw a little bit of change, and some of it was just time or dosage related. And then with ammonia, which was closely related to the methionine based on the NH2 group that it comes from the amino acid group, we saw some time effect. We also saw some dosage effect. Per discussion, so what were the arguments that we thought we could bring out of this study? Those things that we thought maybe we could learn from here. So we saw that increasing the methionine intake could also increase plasma homocysteine levels, which we saw from the first graph or first figure that I showed. You saw the increase from 10 milligrams going up to 100. And then we also observed that increase in homocysteine based on the oral intake of that methionine supplementation was significant when we had the 91 milligram supplementation. And based on all that, the intake was related to the low level of the low observed adverse effect level, and whereas the 46 was close to the no observed, because we really did not see any changes that affected the body, but it was just either there was nothing or it was just low. And continuing with the discussion, so we suggested that if we increased for 4 weeks, there will be a significant tolerance, or it will show actually some tolerance problems. So at this time, we started thinking that, okay, let's go ahead and measure all the other things. So we did go to measure quality of life, cognition, all those, and some of those biomedical variables that were related to that high level, just to see those things that the 100 milligrams could affect. And after looking at all that data, we decided to make a few conclusions. And one of those was saying that the no observed adverse effect level, which is the NOAEL intake, which is the 50 milligrams per body weight, did not have any change. So if you took 46.3 milligrams, it would not really cause any toxicity effect or any tolerance effect in the system. But if you took 100 milligrams, then you could see some changes, but the actual adverse effect was very low. So comparing the 50 milligrams where we saw a drastic increase from 50 to 100 between supplementation time, we saw that the effect at 50, there was really nothing. And then at 100, the level of adverse effect was too low based on the measurement. And then at this time, we concluded that the upper level of safe intake of methionine should be set at 46.3 milligrams per body weight per day. And then the 100 or the 90 would be at 90 milligrams per body weight per day is just based on actual intake on the subject. And this is also all independent of gender, because the females saw a different. If we put all the data separated between females and males, that's the one thing that we technically did not do. But if we group the 6 females that we actually studied and then the 6 males that we actually studied, we saw that the changes was a little bit significant on the female side based on their response then. But we did not go further to measure other things like hormone levels that could be related to these changes. All right. All right, I want to thank these people that we worked this project together in my lab. And that is the director of the lab, Mick Dudes. Marielle is the co-director. And Gerdine, she's now in the Netherlands. She's an adjunct. And then these 2 people that help us with the financial and also with the writing of the paper, and I who coordinated the study from start to finish. Do we still have time? All right. I think that will be the end. Okay. So time for questions.
I stepped out a couple times. You may have mentioned some of this. Just a couple questions.
Mm-hmm.
How many subjects did you have per group?
Yeah, we had 6 females and 6 males, but we initially started with 16 subjects. Then one of them dropped out prior to screening. It was a screen failure.
So was it the exclusion criterion?
Yes, part of it was exclusion because 2 of the other subjects that dropped out during the study was based on exclusion after we had screened and found out that they were not eligible because one of them actually had COPD, but we wanted healthy. Mm-hmm.
So male and female?
Yes, male and female, 6 males and 6 females.
I have a couple more questions, but I'll let this distinguished group chime in here. You have anything?
Yeah.
What were some of the adverse effects that you were seeing?
Yeah, that's a very good question. Technically, we did not observe any adverse effect except those complaints that were reported by the subjects. If they said they had a little light headache, like one of the ladies said she was feeling a little bit dizzy when she consumed, when we went to 50 milligrams, that was one of the things that we observed. But she later on dropped out because we thought if she kept complaining and then we called her in, it was checked by a nurse, and she said she had the same feeling the next morning when she actually took the capsules. So she ended up being a dropout after the 50 milligrams.
So based on his question, you took baseline measures to determine methionine levels.
Yes, we took baseline level.
What were their levels baseline?
The level was approximately close to 10 milligrams. If we go back to that.
You may have gone through that.
Yes. Let's go back a little bit here. Yes. So baseline level was approximately closer to even when we supplemented 10 milligrams. So at baseline, they were about 10 point something, the same thing at 10 milligrams. We only started observing significant changes when we went to increases when we went to 50 and 100. Mm-hmm.
So relative to that one subject that was feeling lightheadedness, dizziness, could it possibly be attributed to the fact that her methionine levels were higher You just didn't adjust to that?
Well, good question. She never came back in, so we can retest her and collect samples to measure. But we also thought that maybe it was also related to some other things. Because when I went back to look at her in her binder to find out what was going on, there were other little things that she used to have in the past. So related to like migraines.
Some people respond differently to
Yeah.
the product.
Yes.
Another question?
Why did you pick that specific age group?
Yes. Yeah, very good question. The reason we did that was because one of the main reasons, number one, our lab is focused on studying older people or aging people. That's one main reason. It might be a little biased for us. The second main reason, what those things that we intend to see during metabolism, the changes are really fast as you age. So like with younger, healthy, and stronger people, there's really less chance for some type of changes to occur in terms of metabolism. But with older people, metabolic pathway changes depending on your dieting and your exercise level or your activity level. Did I answer your question?
Yeah. So, what would be the purpose of the aging population supplement?
Well, one of the main reasons was to see if we could increase their protein synthesis. So because we supplemented them with methionine by injection, and we also gave them the capsules because, number one, it is the start codon for protein synthesis. Then it's also going to help in making other precursors that are responsible for synthesizing other proteins. It could be internal or just muscle mass wisely. But we did not see any changes in muscle mass based on that, so we could not conclude on that side. But we saw increases in concentration of homocysteine level, which is a precursor of cysteine and glutathione, which are antioxidants that could help in the body. Mm.
So you performed the DEXA?
Yeah, we did.
What was the time frame of the study?
The time frame for the whole study was 6 months. Okay. So we did 4 weeks of consumption for each intervention and 2 weeks of flush or washout between each intervention.
So that should have been enough time to depict any lean mass change, right?
If we took if we compared baseline and the 100, but we only compared each intervention time point because we said we would do from the start of the 10 milligrams to the end of the 10 milligrams and then flush out. They came back, we did another DEXA. But if we went back and did start of the 10 milligrams and then the end of the 100 milligrams, then we'll see some changes. Mm-hmm.
So you did our famous food logs probably, right?
We did not do food logs and this because of time.
Okay, because their endogenous diet.
Yeah, that's true. Yeah.
Nobody in here is familiar with food logs.
I think she is. Yep.
How do you think a resistance training program during this study might have affected it?
It could have been. We have a study now that we are supplementing with other amino acids that we are also doing training in it, or we're doing exercise, and we have been seeing some changes. So I bet you if we repeated this same study and included some exercise in it, there will be some great changes. Mm-hmm. We have seen in other studies that we are doing. No, we did not. We wanted to just, yeah. No, we did not. But we do in some of our studies. That's where I just put the fat mass and the fat-free mass, those things that we thought could be directly affected by the supplement.
So the women that were in the study, were you done? I'm sorry.
I was just going to ask if you looked at if you saw any age changes in the blood.
No, we did not look at that.
So the women that were in the study, what were their age range?
The women, they were between 40. I think the oldest person in the group was a woman, if I could remember very well.
You guys throw that word old around too much.
Mm-hmm. Yeah.
It hits close to home.
Because the youngest was a male. I think he was right around 40, 41, 42.
Based on what Ryan
Mm-hmm.
If they're premenopausal, menopausal,
Mhm.
that obviously has an effect on
Yes.
bone mineral density, et cetera.
Yes.
So,
And that's also another thing that I mentioned based on because looking at the female side of the consumption and then not taking into if I account the different type of hormones that they are going through and menopause, of course, those that were much younger, less than 45 years, between 40 and 45, and those that were 45 and older, that's already gone into menopause. I think that's one thing I can go back and look to see if there were any changes between those two groups.
I had just one more question, sorry for all these questions.
Oh, that's very fine. It's making me,
Methionine is an essential amino acid.
yes, sir.
Which means the body does not produce that.
Yes,
Which means
You-
only through our endogenous diet.
Yes.
Were there any recommendations made to the subjects how to attain this rather than a supplement just through a healthy diet?
yes. Some of these things we gave them personal advices, what type of things like nuts they could consume or vegetables and chicken will have about a lot of it. And then they could also buy methionine over the counter if they wanted. The reason, that's a good question because one of my subjects came back to me and told me that it actually helped him, especially when he went for urination. So I was looking, yes, I went back and actually methionine helps to improve with if you have urinary tract infection.
Because when it's all said and done, when we conduct this research, we want to implement it. Hopefully it's going to improve quality of life.
Yes.
So sometimes we just do the research and send them on their way, but hopefully with some recommendations.
Yes. He actually came back to me and asked how he could get. So I went to my boss and asked if we could give him because we still have excess that we did not use. But we ended up recommending that he can go get it over the counter.
Any other questions? Well, thanks for your time, sir.
Very much.